This presentation by Johnathan Craig from American University is from the 7th Annual Clean Virginia Waterways Stormwater and Plastic Pollution Workshop.
A graduate student at American University, and he's going to be sharing some of his research on microplastics and polycyclic aromatic hydrocarbon abundance in Jug Bay region of the Patuxent River. I'll let you take it away, Johnathan. Thank you.
Thank you for that introduction. Hello, everyone. As Zach said, my name is Jonathan, and I'm currently a graduate student here at American University in the Environmental Science Department. And today I'm presenting my research that I performed over summer of 2023 at Jug Bay Wetland Sanctuary, where I looked at the abundance of microplastics and something called polycyclic aromatic hydrocarbons in the Jug Bay region for the Patuxent River. And I did this with my advisors here at AU, Dr. Belaser and Dr. McAvoy, along with Dr. Delgado, who is the superintendent scientist at Jug Bay. So a quick overview, just going into an introduction and background on microplastics, what they are and kind of why we should care about them. And then also what are polycyclic aromatic hydrocarbons or PAHs. Going into materials and methods, so talking about how we look for microplastics and the different techniques we use, going into the results that we found, discussion, and then conclusions and future work that we have left to do with this project. So a quick introduction to the pollutants of concerns that we were looking at. So you probably have heard microplastics in the news. They've gotten a lot of buzz recently, but what is an exact definition of a microplastic? So microplastics are generally defined as plastic particles that are less than 5 millimeters in size or length. And they're usually from primary sources. So those are things such as microbeads that you would find in personal care products, or pre-production plastic pellets, or they can be from secondary sources. So secondary sources is when really large pieces of plastics enter our environment. They're exposed to harsh environmental conditions such as UV light, temperature, mechanical abrasion, that causes them to break down into smaller pieces, and those are known as secondary microplastics. And here I have a few examples of some microplastics actually found in the Jug Bay region. So they're pictured on a nitrocellulose fiber filter plate. And some of the characteristics that we can use to identify plastics is a really bright color. They kind of stand out from the background of the organic matter. And why should we care about microplastics? So microplastics can be ingested by organisms and then work their way up in the food chain of human exposure. There have been numerous recent studies about the health effects of microplastics, both on organisms in the environment and in humans. Microplastics have been found to cause everything from inflammation, oxidative stress. In 2023, a new disease was released in seagulls called plastirosis, which is the accumulation of scar tissue in seagull stomachs from consumption of microplastic particles. We still have a lot of gaps in knowledge about the exact effects of microplastics, but from what we have done so far, we can see that they're not the best both for the environment and humans. One of the other issues that microplastics presents is they're a really complex pollutant. So they can have multiple different polymer types, from common polymers such as polypropylene or polyvinyl chloride. But they can also have a lot of additives. So that can include things such as plasticizers, antioxidants, stabilizers, flame retardants, and those can include things like phthalates, which have been seen to cause endocrine disruption both in humans and in life out in the environment. They also can have a variety of different sources. So I kind of already touched on primary versus secondary. But as such, they can come from a variety of different sources, which makes source tracking a little bit difficult. They have a very large size range, and recently literature has started to talk about nanoplastics, which present a whole new challenge because they are essentially really, really small plastics that are sometimes hard to detect. They have different morphologies, different colors, and another big issue is that they can even absorb contaminants to their surface. So that can include things such as DDT, PCBs, PAHs, heavy metals. As microplastics move throughout the environment, they are able to absorb these contaminants to their surface and then leach these contaminants into the surrounding environment, which can cause a whole range of different effects. And also, microplastics in freshwater environments are of particular interest because it is estimated that around 70% of marine microplastic or plastic debris has terrestrial origins. So understanding how microplastics in freshwater environments, their sources, and how they move through the environment can help us better understand how they arrive in marine environments and help us with solutions to combat this issue. A pollutant that you may not have heard much about are called polycyclic aromatic hydrocarbons, or PAHs for short. So they are a type of organic pollutant that are known to be persistent in the environment. They are hydrophobic, which means they don't really like water. As such, they can accumulate in organisms and bioaccumulate up food chains, which eventually leads to human exposure. The EPA has released a list of 16 of specific PAHs. In this study, we are only looking at 3, which include phenanthrene, fluoranthene, and pyrene. And these PAHs are listed on that EPA priority list. These PAHs have been found to be absorbed to the surface of microplastics in studies in literature. So why are they of environmental concern? Besides being persistent organic pollutants that can bioaccumulate in organisms, PAHs have been found to have a variety of different effects depending on the specific PAH. But the ones that we looked at in this study, there have been research showing that they can be acutely toxic to freshwater organisms and benthic invertebrates. They can also have mutagenic and carcinogenic properties. And there have been some studies linking the synergistic effects of co-exposure of PAHs and microplastics, that the co-exposure of those two pollutants can cause increased bioaccumulation or toxicity effects of each pollution. And their sources are mainly from the incomplete combustion or burning of biomass or fossil fuels. They can also be from impervious surface runoff from asphalt. And then they can also occur from atmospheric deposition, also after the incomplete combustion of biomass or coal. Once in the environment, they can cycle throughout the environment. Sediments are known to be one of the final sinks of PAHs. They can readily associate with the organic matter in sediments, and as such, sediments can serve as their final sink, but they can also be resuspended during increased flooding or higher flow rates in the river too. So a little bit about the area that we conducted this research. So Jug Bay Wetland Sanctuary is located around 23 miles outside of DC, and is located along the Patuxent River, and it is home to a variety of different plant and aquatic life. It is part of the Chesapeake Bay Sentinel Site Cooperative and the Maryland Chesapeake Bay Natural Estuarine Research Reserve. And as such, it is located on the Patuxent River. The Patuxent River itself drains into the Chesapeake Bay and is considered one of the main tributaries to the bay. The Patuxent River has been dealing with a lot of issues in relation to sediments and nutrient overload, but the documentation of microplastics and PAHs has not been done before in the Jug Bay region, and can help provide a future insight to how these pollutants could be affecting the Jug Bay area life, plant and aquatic life. And then in this figure here is just an outline of the sanctuary and its location into the Patuxent River drainage basin. And then, of course, Maryland, Washington, D.C., and Virginia. And then this is a really nice photograph of an overview of Jug Bay Wetland Sanctuary. So some of the main research objectives and hypotheses that we had for this project, the main goals that we had were to determine concentrations of microplastics in water samples in Jug Bay, since this hasn't been recorded before, and of course take note of any qualitative characteristics such as color and shape, and then examine relationship between water quality parameters and microplastic concentration. Microplastics in freshwater systems is an emerging area of research. As such, there is not much information about how microplastics interact with different water quality parameters in freshwater environments. So we're hoping to kind of gain some insight into that. And then assess spatial and temporal differences in microplastic concentration and water quality parameters. Since microplastics are ubiquitous throughout the environment and found in all different environmental media, one of the hypotheses is that the microplastic concentrations would be above zero in water samples. And then we had two stations, and one of the hypotheses is that on the main river, microplastic concentrations would be higher than those at Station 2, which is a tributary located in the marsh. And then that PAHs would also be detected in sediment samples collected. So getting into the materials and methods, so how we were able to gather the data to test these hypotheses and to understand these research objectives. So we used two stations. So station one was located on the actual Patuxent River at the end of a lookout. And then station two was located in a small tributary that is in the more marshy wetland area. And for a total of 10 weeks, we took water samples at each station, station one and station two, 2 liters of water samples. And then we took sediment samples at week 1, week 5, and week 10 of sampling. Water quality parameters were also taken alongside water samples and for microplastic concentration, and those included things such as dissolved oxygen, pH, salinity, turbidity, flow rate, and we even looked at depth and precipitation too. So in order to better understand the water quality parameters, we used a variety of instruments. So a YSI probe allowed us to understand better the pH, temperature, pressure, dissolved oxygen, a turbidity meter to measure turbidity, and a flow rate meter to measure the flow rate of each site. When we were collecting samples, we used glass jars and tin foil to avoid any plastic contamination into our samples. Glass jars were used to collect the water samples, and tin foil trays were used to collect the sediment samples that were air dried in our lab. And like I said, this just helped us prevent any plastic or microplastic contamination. A lot of our clothes nowadays, or a lot of containers are made from plastic, which could introduce microplastic contamination and cause our samples not to be representative of the microplastic concentration that is actually found. So for analysis of our samples, we use a filtration method. So essentially, the water samples were run through a vacuum filtration system, and we filtered using a 0.45 micron filter size nitrous cellulose fiber filter. That just allowed us then to take that filter under a light microscope to observe any microplastics. So I have another example here of a microplastic that was found on the filter plate. So we, alongside recording the amount of microplastics, we recorded color and microplastic morphology, so fibers or fragments, to better understand and characterize microplastic presence in the Jug Bay Wetlands Sanctuary region. And some of the criteria we used is that microplastics under a microscope, besides having really bright colors that kind of set them out and distinct them from the background, they normally are smooth and uniform, and kind of have like an unnatural shine or sheen to them compared to like piece of organic matter or something. And to avoid further contamination, we made sure to wear cotton lab coats, gloves, and leave out air filters to account for any microplastics that might be in the air that could be contaminating your samples too. Organic matter analysis was done using loss of ignition method. Organic matter analysis was conducted in order to better understand the relationship between organic matter percentage and PAHs. PAHs, like I previously stated, can attach to organic matter in sediments. And in research, it is found sediments with a higher amount of organic matter percentages tend to accumulate PAHs. So a loss of ignition method was used just to better understand the organic matter percentage of the sediment samples that we took. pH extraction and analysis. It was a lot of steps, but essentially a quick overview is that we took the sediments and using dichloromethane, we were able to extract organics from the sediment and then separate them into a PAH and alkane fraction. And then those fractions were eventually run on a GCMS, which allowed us to detect any peaks that might correspond to PAHs that would be in the sediments. So kind of getting into the results and discussing what they mean in the context of current literature. So microplastics were found at station 1. The average microplastic concentration was 4.81 microplastics per liter. The dominant microplastic shape was fibers, with over 84% of total microplastics having the fiber morphology. The dominant color for fibers was black, and fragments were found at station 1 with the dominant color of fragments being blue and orange. And then over time, there was a negative decrease or correlation in microplastic concentration for sampling. And overall, at station 1, black fibers were the most commonly identified type of microplastic that was found at station 1. And then looking at station 2, so microplastics were also found at station 2 with an average concentration of 6.60 microplastics per liter. The dominant microplastic shape was also fibers, with a really higher percentage of fibers, 94% of total microplastics having the fiber morphology. The dominant colors for fibers was black, and the dominant color for fragments was blue. And same at station 1, the most dominant type of microplastic found was black fibers. And also similar to station 1, there was a decrease in microplastic over time of the sample. So kind of looking at the results of station 1 and station 2. So to test one of our hypotheses that the station 1, which is on the river, would have a higher microplastic concentration, a one-sided t-test was run. Unfortunately, there was no statistically significant difference between the means of station 1 and 2. But there were differences in certain parameters of water quality between station 1 and 2. So dissolved oxygen, salinity, and flow rate differed between station 1 and 2. This kind of makes sense since station 1 is fast moving water on the river. We would expect it to have a faster flow rate and also to have higher amounts of dissolved oxygen compared to the stagnant water at station 2. And then station 1, we also expected the salinity to be different. With faster moving water, the salinity was lower compared to the salinity at station 2. And both stations had fibers, and specifically black fibers, as the dominant microplastic morphology and color present. So to better understand if there were correlations between our microplastic concentrations and these water quality parameters, something called Spearman's correlations were run. This essentially allows us to see if microplastic concentration is correlated to a specific water quality parameter. So at station 1, it was found that microplastics had a negative correlation to turbidity. And then at station 2, microplastics were found to have a negative correlation with water temperature, but a positive correlation with flow rate. And essentially, this provides us with better data to understand how microplastics interact with different environmental parameters, which can be useful in environmental modeling situations or better understanding how microplastics are transported in freshwater environments. Now to the organic matter and PAHs. So some good news, PAHs were not detected in any of the sediments analyzed, or they were below the detection limit for our GCMS. And then organic matter percentage was higher at station 2 compared to station 1, with organic matter decreasing over time. This may be due to the fact that station 2 was in a more densely vegetative area than station 1, which was more in the river. And decreases in organic matter may be due to increases in temperature, which causes rapid decay of organic matter in the sediments. So kind of putting that in context of current literature, what does all of this mean? So looking at different study sites and locations, so Jug Bay Wetland Sanctuary is considered a more pristine environmental area that has been the site of many efforts for environmental protection and conservation. As such, compared to other freshwater environmental rivers around the world, such as the Ottawa River in Canada, our concentrations were higher, but they were not the highest concentrations that I have seen in literature. So for example, in really heavy urban environments, microplastic concentrations can get upwards of 300, 400 microplastic particles per liter in really heavily polluted urban areas. But because of the ambiguous presence of microplastic throughout the environment, it is not uncommon to find microplastics even in areas that are considered pristine or isolated from very densely populated areas. And then kind of looking at the relationship of water quality parameters to microplastic concentration. In literature, flow rate is mostly negatively associated with microplastic correlation. This is kind of opposite of what we found in our study. But fibers being the most common morphology does track with what is presented in literature. Specifically, wastewater treatment plants in their effluent water that they discharge. A lot of wastewater treatment plants are not equipped to deal with microplastics. As such, a lot of wastewater treatment plants' effluent water have microplastic fibers in them. And not surprisingly enough, just upstream of our sampling site, there is a wastewater treatment plant that does discharge water upstream into the Patuxent River of our sampling site. And temperature is normally negative correlated, which we found. Turbidity normally has a positive correlation, which we found the opposite. And this kind of speaks to the complexity and the fact that microplastics in freshwater systems are an emerging research field. Because you could search literature and you'll have some studies saying one thing and other studies saying another, because we're still kind of better understanding this pollutant in freshwater environments and how it exactly is interacting with different environmental parameters. That's also to say that there are a lot of other factors that could affect microplastic concentration, things such as population density, land use, slope or elevation of the catchment area, all of which can have effects on microplastic concentration out there. But with this study, it allows us to give a baseline kind of understanding of the presence in Jug Bay and better understand the transport of microplastics from freshwater systems into marine environments. So kind of wrapping up with conclusions and future work. So microplastics were found at both station 1 and 2. Station 1 having an average of 4.81 microplastics per liter, with station 2 having an average of 6.60 microplastics per liter. The dominant shape was fibers, and the dominant color of microplastics was black. Spearman's correlation revealed that certain water quality parameters could be correlated to microplastic concentration. Turbidity had a negative correlation to microplastic concentration at station 1. Flow rate had a positive correlation to microplastic concentration, and water temperature a negative correlation for microplastic concentration at station 2. PAHs were not detected in any of the sediment samples that were gathered or were below the detection limit. And this study allows for further research studies to be conducted to determine the exact source and distribution of microplastic pollution, both in Jug Bay Wetland Sanctuary and the Patuxent River as a whole. And this also allows for a better understanding of microplastics in freshwater systems, their presence, and possible transportation in freshwater systems. So we still have some work to do on this project. So future work is the sediment samples that we collected. Microplastics will be extracted and identified, and then chemical analysis will occur for both the microplastics found in sediment using a technique called thermal deposition pyrolysis, GCMS. And then in collaboration with the University of Florence, using a machine called an FTIR, chemical analysis for filter plates will be performed. And this will give us a better understanding of the exact polymer types and any absorbed contaminants to the polymers or slash microplastics that we found in the Jug Bay Wetland Sanctuary area to better understand their sources and possible environmental impacts of these microplastics too. And with that, I would just like to thank both Jug Bay Wetland Sanctuary for this research opportunity, AU for this opportunity to perform this research, and thank you to the Clean Virginia Waterways for the opportunity to present my research at this conference. It's always a nice opportunity to talk about my research, so thank you for this opportunity too. And with that, I'm all finished, and if there are any questions, comments or concerns, I'd be happy to answer any. Thank you.
Thank you, Jonathan. That was awesome.
Thank
Do
you.
we have any questions in the room? Katie, if we have a question online, we can take one question while I get the next presentation set up.
Yes, we do. Somebody has asked you, Jonathan, does the literature say anything about why fibers tend to be more commonly found as opposed to, broken down pieces from larger items? Is it that they're more prevalent in the water column or in all medium?
There's a couple reasons. So, one of the reasons that fibers are mostly common in freshwater environmental systems is from wastewater treatment plants. So a lot of our clothes or textiles are made up of synthetic polymers. As such, water from our washing machines, when they enter wastewater treatment plants, those fibers are not able to be properly filtered out. And as such, when the discharge water enters the environment, it can have a really, really high concentration of these fibers that is then dispersed into waterways. Also, it does deal a little bit with densities. So fragments are more likely to sink down in sediments. This is fragments can gather like biofilms or other pollutants that cause their density to increase and sink. Fibers normally have lighter densities, so those are normally common on the top surface water where our sampling occurred. But due to different densities of different polymers, you can find in a complete water column a whole bunch of different microplastic morphologies due to all their different densities. So there's like a couple reasons, but I would honestly say that probably a lot of the times microplastics, microfibers normally come from wastewater treatment plants discharge, because a lot of our wastewater treatment plants in general don't have a lot of technology to combat microplastics to filter them out.
Great, thank you. And I guess one last question if we have time, Zach?
Yeah, we can do a quick question.
Okay. Somebody asked about protocols. Are there published protocols for the research you did to determine the type of microplastics you were finding in the water column?
Yes. There's a whole bunch of out there, but we did follow protocols for contamination controls. That's one of the biggest ones to prevent any external microplastic contamination, whether it's from the air or our clothes. So our contamination protocols were found in literature. Also in literature for identification of microplastics. So there are a whole bunch of research articles talking about kind of a standardized categories for microplastic shape. So things such as fibers, fragments, filaments, foam, pellets slash granules. So we did try to follow for identification of the shape morphology and color, what is presented out in literature. And then same for the rest of our protocols with filtration and the hopefully future chemical analysis. Those are kind of commonly used techniques. It is a little hard because with this emerging area, if you search the literature, there's a lot of variation in different techniques that people use, from visual identification to density separation to a whole variety of different things. But we try to, given the resources that we had here at AU and what is presented in the literature, we did try our best to follow kind of laid out protocols in published literature.
Okay, great. And I know that we're running behind, but a really quick question. Hopefully this is a yes, no. In your literature review, did you find anybody who's looked at the sources of these microfibers? Are they mostly coming from clothing, textiles? Because we know there's nylon ropes being used in the water environment and tarps. So did you find any research where people are trying to break down the sources of these fibers?
I do know there's research in marine environments, understanding whether the microfibers are coming from like fishing nets or fishing activities compared to transport from terrestrial. There has been some research at looking at wastewater effluent water to better understand the concentration of microfibers entering our water systems and their polymer composition. So yes, there is a lot of ongoing research to understand better where these microfibers are coming from slash microplastics, and to better understand their sources too. Because a lot of policy now is kind of focused on source reduction because microplastics are very tricky to remove from the environment. So if we can better understand how these microplastics are getting in these freshwater environments, we can better reduce their sources. So a short answer would be yes.
Okay, thank you so much. Great presentation.
Thank you.
I'm going to turn it back over to you, Zach.
Awesome. Thank you, Jonathan. And if anyone has more questions for him, you can reach out directly to Jonathan.